So thank you everyone who are tuning in today. Welcome to EMBL's final webinar during our Open Access Week in 2022, Open Science for Climate Justice. My name is Victoria Yan. I'm the Open Science Specialist at EMBL. So in the spirit of this year's Open Access Week, Open for Climate Justice, we have invited 4 panelists, Tony Burdett, Sarah Dyer, Thangot Ebenetzer, and Peter Harrison from EMBL-EBI, to discuss open genomic data and open data infrastructures that can help us to adapt to the imminent challenges posed to food security and biodiversity. They will discuss very fascinating projects that will enhance the usability of genomic research, strengthen collaborations, and build capacity to generate, analyze analyze genomic data on an international scale. Our webinar will be comprised of 40 minutes of panel discussion. And following that, we will have 20 minutes to answer the audience questions. So if you have any questions, please ask your questions in the Q&A. If you would like to ask the questions yourself, please wait until the Q&A session, raise your hand, and we will ask you to unmute. So without further ado, I would like to hand it over to our first speaker today, Tony Burdett, to introduce himself and his work with FAIRplus.
All right, thanks, Victoria. I'm going to share my screen quickly just because I have a few slides just to sort of introduce some of the concepts that I'm going to be talking about. So I think the first thing to say is that I'm not specifically involved in some of the sort of initiatives that Victoria talked about for climate justice. So I'm not directly participating in some of the food security or global biodiversity initiatives that I think Sarah and Peter and thank God might talk a little bit more about later. But the project that I am involved in is very heavily focused on generating fair data and actually helping organizations, projects, and people understand how to better share their data openly. And we talk a lot about fair data, and I will talk a little bit more about what that means. But essentially, when we talk about fair, we talk about findable, accessible, interoperable, and reusable data. And really, the R is the important one here. How can we make sure that of the wealth of data that's out there that's being collected, we make it as reusable as possible? And this is important because we know that we know that sort of data science is a really core element. I mean, I talk about and work in the health research and innovation space quite a lot, and the FAIRplus project that I'll talk about in a minute is heavily involved in this kind of health research component. But really, data science is becoming increasingly important in biopharma research, and likewise the impact of AI and machine learning techniques is growing rapidly. And the really critical thing to help these sort of data-centric approaches for research improve is making sure that we have data that computers can understand. And quite often our data generation efforts don't support this high level of reuse. We sort of tend to leverage our data for a single research project rather than sharing it more widely and pumping it into these sort of global initiatives that we might talk a bit about today. And this pie chart here sort of really emphasizes that problem, that we find that data scientists in general spend sort of 80% of their time on these 2 main activities: cleaning and organizing data, and actually just collecting the data that's useful to them. So this 80% of the pie chart here is actually collecting and cleaning the data. It's also the bit that no data scientist ever really wants to do. It's not the fun bit, like finding out, making new findings, deriving insight from the data that's already out there. It's just a curational activity. So, part of the importance of fair data is reducing that 80%, and there's a huge opportunity there. The other thing to call out, I think, is that fair data, we talk about a lot, it's not necessarily open data. The A in FAIR stands for accessible under well-defined conditions, which might not mean all conditions. So there might be legitimate reasons to shield parts of the data and services, especially depending on where the funding comes from. There might be personal privacy issues. I think in the global biodiversity and sort of food sustainability questions, the opener we can be is better. So, you know, I ideally don't want data sets locked away under some sort of access restrictions, but, you know, the important thing to bear in mind when we talk about FAIR is it's not always equivalent to open data. So the project that I work on is called FAIRplus, and this really sort of tries to dive into these main sort of challenges. We know that data is not reusable at scale, and that's ultimately the ambition behind FAIRplus. There's that reuse at scale has sort of four main questions or four main challenges that drop out of it that we've tried to work with to sort of address within the FAIRplus project. So there's no practical advice on how to do verification or make your data more accessible, more reusable. There's some resources that we've developed to help with that. Often organizations, people don't know how fair their data is already. And that's a question that we would like to try and resolve. And we have sort of processes in place to sort of assess how fair projects and data is. Developing the steps to become more fair can be difficult. So we've designed processes and a maturity model to help with that. And often we find that we hear people say that, you know, my organization doesn't care anyway, as long as we've got our paper out at the end of the day, it doesn't really matter. And we really need to think about changing the data management culture within organizations, projects, and within our communities more widely to make sure that this really valuable insight that can be derived from data that's been collected, that's out there in the world, can be taken advantage of. You know, I think there's sort of everyone on this webinar, everyone that we work with has an opportunity to sort of try and contribute to this data management culture change, and encourage people and your organizations to do a little bit of investment in sharing data openly, because it really does feed into like really wider global impact. And the other thing I wanted to call out in my introductory slides is this highly collaborative approach, right? This idea that we want to share data more openly and understand how to capitalize on the sort of reusability of data takes a whole village of people, right? It's a team sport, and making sure that we can unite people who understand data, who are data scientists, have really deep expertise in sort of data management initiatives, as well as understand the science that's generating the data and the way that the data wants to be reused. You can't hold it in one person's head. It's a really collaborative initiative, and we have this kind of approach in FAIRplus where we define these sort of cross-functional teams, and we call them squads, that bring data scientists and bioinformaticians and biologists and researchers in the clinic together to sort of identify what the really important things to pull out of the data are, how we can facilitate reuse, and how that can benefit society more widely, as well as the people who can actually practically go and change the way things are done, whether that's changing what database you use behind the scenes, or sort of, you know, picking an ontology to use in the data. And then how does this sort of carry forward? You know, we actually then sort of try and practice these techniques when we get data that's deposited to EBI archives. So one of the databases that my team's helped to deliver is the European Nucleotide Archive. And we invest a lot of time and energy in working with projects, some of the projects shown at the bottom of this slide here, like the Darwin Tree of Life, that are depositing data with us. We spend time and energy working as an organization, working with those communities and helping them to practice some of these techniques that I've talked about, to encourage them to share their data more widely in the most reusable way possible. And I'm pretty sure that Sarah and Peter will talk more about this as we go forward. So that's all I wanted to say by way of introduction. I guess I'll hand over here. So and we'll take sort of throw it open to discussion later. So thanks.
Thank you, Tony. So next we have Sarah Dyer.
Hi, everyone. Thank you for the invitation to be here. And thanks, Tony, for that great introduction to FAIR data and the FAIRplus project. It's really cool stuff. So I'm part of the Ensemble project at EMBL-EBI, as is Peter Harrison. So you might hear more about that from him also. The Ensemble project is a kind of knowledge base and a platform where we bring together genomic data from a variety of species, and we run standard processes and we present that back to the user. So we have a genome browser to allow people to come in through the website and look for genes of interest, for their species of interest. We run a lot of comparative genomics pipelines so that we can compare or present a gene and how it's conserved kind of across the tree of life. And we also bringing open data from a variety of sources. So I guess the first thing to say is that we're an open platform, the code is all openly available, and we use public data in the platform. So there are various different bits of ensemble. You have the kind of vertebrates, which is the traditional, the original part of the project was focused on vertebrates. My part is focused on plants, and I also have the metazoa division. So that's all the non-vertebrates, a lot of invertebrates, a lot of insects going in there. I'm also responsible for our outreach team. And then I have connections with worm-based projects as well. So a bit of a range of things, but my kind of the thread and area of real interest for me is agriculture. And so how plant genomics can support agricultural research efforts and improved breeding. And so of course, it's very important thinking of food security. And that's why I'm really excited to be here today, talking about this in the context of climate and food security. So we work with communities to kind of bring data sets in when there's been, particularly plant genomes tend to be very large and a lot of effort has gone into generating them traditionally. So when they get deposited in the archives, like in the ENA, then we can bring those genomes in and we can layer on gene annotations and different data sets and link out to other data resources that are also open that people can then go and explore and learn more about gene function in the kind of areas of interest. If they have a particular area of the genome they're keen to know more about, they can kind of use our tools to go and explore the proteins that are expressed and connect out to get the kind of function for those. So our users are a mix of, well, kind of, I suppose, many molecular biologists, molecular geneticists, some breeders. And so that's how we have, we kind of, when we're bringing in data sets, we think about who we're going to kind of reach out to. So agriculture is a big one. Of the more than 100 genomes that we have in ensemble plants, the vast majority of them are agricultural species or model organisms. And so we bring those kind of crop genomes in and then support our communities through our help desk and outreach and training activities to be able to query those data and find useful knowledge and generate hypotheses that then can go on and be tested further downstream. So I think I'll stop there and hand over.
Thank you, Sarah. And I'd like to ask Thangout to talk about your work with the African Biogenome Project.
Okay, so thank you very much, Victoria, and for inviting me. So my name is Thangout Ebenezer, and I'm a bioinformatician at with the Universal Protein in the Universal Protein Resource team, and I'm helping to initiate the African Biogenome Project. So basically, there are 3 goals within the African Biogenome Project. The first goal is to coordinate all non-human genome sequencing activities going on the continent. And the second goal is to initiate new ones. And then the third goal is to then see how that can be deployed into regional infrastructure, such as or agencies such as the Africa Union Inter-Africa Bureau for Animal Resources and also other plant agencies on the continent. And ultimately, how can that be used to inform policies across Africa? So Africa BP started around 2019, 2020, and it's basically the first attempt was to have a public engagement session and trying to get buy-ins from people across the continent. And then it was there was a consensus that this is a good thing to do across the continent. And since then, there has been several series of workshops and engagement. And last year, around June, the pilot project was launched, and the aim is to sequence about 2,000 to 2,500 species. And that again involved bringing together several scientists across Africa, and of course, linking the African scientists with partners in Europe and North America as well. And in early 2022, the Africa BP published its position paper in the journal Nature, where the goals and the big picture was actually highlighted. And since then, we've also spoken about Africa BP in several fora. And in August, one of the things Africa BP did is to kind of institutionalize Africa BP, because the project has started in the reverse order. Mainly, this has been a grassroots effort. So in most projects, you have an institution, and then you have the funding, and then you start the project. In Africa BP case, it's actually the reverse order. You have the proposal, and then you have now look for the funding, and then the institution. So it's been institutionalized within the Science for Africa Foundation. So it's an agency across in Africa that kind of coordinates some of this kind of project. And the idea is to tie it to the Africa Union Agenda 2063, as well as other continental framework. And very soon again, we'll be doing the press release statement. So, and there are basically 4 grand challenges or programs that Africa aims to solve. So the first focuses on animal genomes, focusing on looking at endemic African species and then looking at endemic plant species. And of course, the third one basically focuses on biodiversity framework, issues around the Nagoya Protocol, the post-2020 biodiversity framework, and all of the issues around surrounding access and benefit sharing, which I think would be linked to the discussions today on open access. And ultimately, the fourth one focuses on the Open Institute. And this was set up in response to some of the discussions that have been ongoing since the formation of Africa BP on issues around capacity. And given the fact that the continent of Africa, when it comes to the generation and use of genomic data, the capacity to generate between Africa and the rest of the world kind of differs. And of course, that again hinders the discussion around open access or the progress that will be made. So what that means is that the continent cannot sequence at large scale as is possible in Europe or in the US. And of course, in the same vein, they can also not use the data being generated. I think again, that tends to bring some barriers in all of those things. So the Open Institute aims to see how can we maximize all of those things, within the context of also sequencing genomes and making it readily available. So in the course of discussion, again, I'll talk about all of this and the access and benefit sharing and the partnership with EMBL, and as well as looking at the big picture. Thank you.
Great, thank you so much. Thank God for that introduction. I would like to hand it over to Peter.
Hi, thanks. I'm Peter Harrison. I'm the Genome Analysis Team Leader at the European Bioinformatics Institute. And I've been working for a number of years with communities to support their efforts in distributing open access data. And most recently, we've been supporting global biodiversity efforts. So there's been a real explosion in biodiversity data, both around improvements in sequencing technology, but also sort of a public drive to really understand global biodiversity and climate loss and mitigation. So we've really been working with communities from around the world to really focus on generating open source, high quality reference genomes that are free and free to anyone to access and to use, and really how this will then really support different communities to then be able to access reference quality genomes that they've never had before. So really this is part of the Earth Biogenome Project, which has been described as sort of biology's moonshot to sort of sequence and catalog and characterize all of the Earth's eukaryotic biodiversity. And our role at the European Bioinformatics Institute has been not only to sort of archive and openly distribute this data, but also to try and help communities into finding the data that they're wanting to use, which species they can use for their research, and really to try and gather together all of the different types of data that they can coordinate in a single way and distribute to the community. So one of the things that we do is generate data portals, and these provide a single access point to sort of the wealth of data that you can find for a project. So these are fully open, high quality reference genomes, all of the contributing data that's come towards that. We draw in all sorts of information on climate information that's available, information on sort of scientific interest that's going on in individual countries. And really, within this project, the Darwin Tree of Life, which is aiming to sequence all 70,000 species, eukaryotic species in the UK and Ireland, we're generating this ever increasingly automated and rapid genome engine. So that as soon as data is generated and has come off the sequencing machine, we make sure it's openly available to the scientific community to use with as minimal delay as possible, to really try and reduce the amount of time that people have to gain access to key data. And another key aspect is this: it is a global mission to sequence all of the Earth's eukaryotic genomes. So status tracking becomes really key because species are spread across borders all over the world. So we're really trying to coordinate with lots of projects around the world to ensure that we're not sequencing the same species over and over again, and really trying to get as much benefit to the global scientific community as we can. And really, within the Darwin Tree of Life, we have some of the some amazing sites of special scientific interest, such as Whiteton Woods, which has been incredibly studied over the last 80 to 100 years. And really, this is an exceptionally sort of rich area for biodiversity. And what we're now doing is by providing open access to all of the reference genomes for these species, we're really revolutionizing the way that scientists can conduct climate research in these sites, and supporting scientists with access to reference quality genomes that they've never had before. So really what this is doing is really trying to, through this sort of standardized annotation, harmonizing the way in which we describe the data, as Tony was describing, but also ensuring that we intuitively provide ways that people can openly access the data. We're sort of getting maximum impact from all of the data that we're generating and providing openly to the community to really try and support conservation and climate change mitigation, food security, and then access to biopharmaceuticals and biomaterials.
Thank you so much, Peter, for introducing your work. So just to start with a question for all of the speakers here, who are the end users of your project and data? Can you give us some examples of use cases in the real world for preserving biodiversity, or in the case for Sarah in food security?
Don't know who wants to go first. I mean, the end users of the sort of FAIRplus project that I talked about are sort of, it's a different audience, right? It's a different community of people. We're very heavily involved in target identification for human drugs. So it's a completely different domain. But slightly more generally, I think the sort of users of the outputs of the FAIRplus project, we're really trying to build this model of how to share data more openly, more fairly, which means that kind of any data scientist really could benefit from that. Any sort of data scientist or data manager could benefit from that. So we really are targeting this kind of cross-domain thing. So actually like rolling some of those techniques into different domains like global biodiversity. I mean, I think the approaches that I talked about in FAIRplus and the approaches that Peter talked about in his slides are really sort of very, very similar. And, you know, those approaches are transferable. So, you know, I guess the FAIRplus audience, the users of our project, are really sort of data managers and data scientists who are generating and using data at scale. But in terms of the global biodiversity and food security, I guess I'm going to look to the others to try and sort of tap into that component.
All right, I'll take the next one then. So yeah, for us, it's obviously kind of different than Tony was describing. So we have our lots of plant genomes, agricultural species. We also have insect genomes. So where we have researchers that are looking into kind of, you know, interactions between insects and plants, then that's, you know, we're able to kind of provide the genomics aspects for both of those, which is quite cool. And we also have other groups which represent kind of fungal and other pathogens, all within Ensemble, within the different sites. I guess typical users for us would be biologists that might come in using the web browser to kind of query a gene, or they have a marker and they want to know, you know, they know that this region of the genome is important in like resistant plants, but they don't know exactly what the causes are underlying. So then they can come in and explore the genes and get to the function and kind of, you know, figure out how that works. And look at expression data and those other kind of things that we connect to different EBI resources. And so yeah, that's I suppose one of the main ways that people would come in, but we also provide lots of means to extract data and work more bioinformatically, more programmatically. So we have kind of pipelines for that. We also have a biomart, which allows you to kind of come in and get a big chunk of the data and work on it. So we'll have a lot of bioinformaticians that work with us, as well as being more kind of lab or traditional biologists. And as I mentioned before, we also, you know, because of the standardized data that we provide, we're also a resource that breeding companies will come and kind of take copies of our data, and they will kind of use it with kind of to support their activities for actually breeding different varieties and improved crops.
So, and in the biodiversity world, really, this is supporting, in a sense, it's supporting fundamental science. So what we're providing is open access to reference quality genomes for species that have never had them before. So there are communities that are now able to suddenly pivot their research to be able to utilize genomics data for the first time. And so this is being used in a huge variety of ways. So for example, in the UK, it's being used to sort of support safeguarding of our woodlands, to understand the sort of the biodiversity of our woodlands. How many different species do we have there? How closely related to some of those species? It's being used within bat populations to understand virus transmission in bat populations, particularly. There's some specific projects going on in Ireland to do that. It's understanding the diversity of our coastlines and how that's being impacted by increasing temperatures over time. So there's a sort of a huge array of applications that you can have for understanding issues of biodiversity loss directly, but also climate change. But then there's also a hope that by sequencing more species, we'll actually sort of discover new aspects around biopharmaceuticals. So some of the greatest discoveries we've had in medicine have been from naturally occurring substances that we found and adapted to use for medicine. So by actually gaining a far greater understanding of the biodiversity we have and preserving it before we lose it. So the principle of a biodiversity genome arc, in a way, to preserve the knowledge that we have of the species before we lose them, it really can sort of fundamentally change sort of the very nature of how we're doing biology, which is really the aim of some of the Earth Biogenome Project's goals.
Okay, so I think for within the context of the African Biogenome Project, so there are 2 to 3 use cases. So the first one is on policy, policy framework. In June rather, the African Union invited members of the African Biogenome Project to talk about the project because the African Union did say it in the Nature paper, to talk about the project and see how it can basically link up to the continental agenda on having a biobank and things like that on agriculture, basically across the continent. So that is one use case. One of the things again that would help to do is to see how can that help to improve policy across the continent. Now, of course, we also have use cases from the plant breeding academy and also the animal breeding academy in Africa. And ultimately, again, there are scientists who are also now interested in this. And the fourth one has to do with the post-2020 biodiversity framework. So whereby looking at issues of Nagoya and also the beneficiary, and more importantly, digital sequence information, which kind of represent all of this. So for the continent, there is no large-scale effort to generate sequence, and the Africa Biogenome Project aims to present the opportunity for that to happen, and that will provide the scientific evidence required to engage the conversation on the digital sequence information from the African perspective. And I think these are some of the core use cases. And of course, we would expect that as a project evolves, there will be use cases in the biotechnology, the rooibos plant in South Africa, and also other medicinal plants that are basically endemic to the continent of Africa.
Yeah, thank you. Thank God. That's really interesting to hear from everyone, the wide range of use cases that's made available by these new open access types of genome data. So I would like to ask you, for those researchers who are interested in your projects, how can they get involved?
I'm going to let someone else go first this time. So yeah, I think it's less relevant for me to answer that one.
Yeah, so maybe I can just go first. So there are basically 3 ways to get involved within the Africa BP. And the first way is that we have what we call the partnership framework and also membership. And we're also a partner to the Earth Biogenome Project, which Peter talked about, and also the Vertebrate Genome Project. So the first, we have the institutional membership. So what that means is that we have institutions coming as an entity to the members. We also have corporate membership, and whereby we have members of the corporate world or industry coming together within the project. So at the moment, we have about 40 plus partners, and about 5 to 7 of those are industry members, and the rest are institutional members. Then we also have assured project members membership, whereby, so basically what this does is that there are existing projects on the continent already going either at a small scale or on a different scale. So the idea is to bring everyone together, and as Peter mentioned, within the context of the Earth Biogenome Project, it will be helpful that when a genome is being sequenced, most other communities know, so that we don't have to duplicate effort. And we also have membership within the pilot committee and steering committee. And because at the moment, there are over 100 members within the African Biogenome Project, and about 80–90% of them are based on the continent.
Oh, Peter, you want to go?
Yeah, I can go. So really, all of these efforts are open to everyone. So this is both all of the data that we generate is open to you for you to use as researchers. So if you're wanting to gain access to any of the data, we have it all available from our data portals. So you can either search specifically for the project, so Darwin Tree of Life, or the European level, the European Reference Genome Atlas, or at the global level, the Earth Biogenome Project. But also, if you have a particular interest in a particular species, you can also influence which species are going to be have their reference quality genomes generated first. It's going to be a very long endeavor, probably spanning over more than a decade to sequence all of these species. So if you have access to material, or you know where a particular species is located, people would very much like to hear from you, and really we're always looking to get involvement, and particularly at the European level, it's very much a community effort. So if you're wanting to contribute to the annotation, if you're wanting to contribute to some of the sequencing projects, or contribute effort from your labs, it's very much an open effort, and you can join the working groups and really contribute your sort of scientific expertise to those efforts as well.
I would say from our side, so a great way for researchers to get involved is to deposit your genomes and your data in the public archives, and then we can bring them in. Quick plug for open data. So yeah, if you've generated a genome, some variation data for a species that we already have, and you think we should have it, tell us. We have a help desk that we're always responding to people. We love to hear from our users. So, you know, reach out if there's something that you've read about in an article that you think we should have. Please do make suggestions, and we'll kind of add it to our lists. So yeah, get in touch with us. If it's something larger that you want to do, perhaps you're doing a pangenome, so you would like to have many genomes added in, okay, you know, then we're more than happy to partner in these projects. So that's a way that we have many collaborations on the go. We'll work with research groups and consortiums to kind of bring larger data sets in. And the other way, as I mentioned, I'm responsible for the outreach. So we do a lot of training events. And if you think that you would like to host an event and have one of our trainers come and talk about our different resources, then again, get in touch and we can organize that for you.
Yeah, thank you all. Tony, would you also like to add how can researchers get involved with FAIRplus?
Yeah, I think at this point, actually, the FAIRplus project wraps up in about 2 months. So really, it's actually just leveraging all the stuff that we've done. And there's a bunch of stuff. I pointed at some of it in my slides. Like there's a FAIRplus cookbook that you can use to sort of like look up some recipes about how to do better data management and a maturity model. There's a number of outputs, and there's like the main part of the FAIRplus project has actually been to try and define a framework for fairification. So how do you put the practices in place to ensure that your community, your project, your organization are generating fairer data? And that's the thing that you can pick up and use. Less about getting involved at this point because we're about to wrap up. But yeah, always very willing to hear feedback, I think, as well. So, you know, if you try any of this stuff out and you find that it does or doesn't work for you, then please let us know.
Thank you for that. So it's very interesting to hear from all of your projects that not only are your projects making data available, but they're also providing a lot of expertise that help build capacity for creating and analyzing data amongst many research communities, as well as different types of actors, both researchers in academic setting and also in industry settings. So what are your perspectives on the long-term impact of enabling all of these research communities to adopt open data and FAIR principles in their research?
I can go first on this one, and I noticed that Anne asked a question in the Q&A as well, and I'm going to touch on this when I'm going to sort of hopefully answer Anne's question here, because Anne's question is a great one, right, about whether we've done any work on the CARE principles, aligning them to the FAIR principle. And the answer to that in FAIRplus is no. But what we have done with FAIRplus is create this framework, create this sort of process management structure for capacity building. So that is relevant because we can then actually train people on this model and encourage communities, including sort of those for like indigenous people. And actually, we're working with H3 Africa going forward. We've got a new project in my group, and we're adapting some of these principles to build capacity across a number of different organizations, in this case in Africa, about actually building up capacity for generating and managing FAIR data. So this kind of component that says, actually, we've produced this sort of framework and we want to train and teach people how to use it, it gives them greater sort of control, greater agency of managing their own data in a way that's more interoperable, more fair, you know, without necessarily going through the process of saying, I've generated some data here, EBI, you take it and clean it all up for me and then store it in a big centralized database. We actually create the environment where more people can retain ownership of more of their data, but know that it's part of a sort of global community of interoperable data, which is of course the goal of sort of open data. So you know, I think this capacity building component is really important. It's something we've been very strongly focused on within the FAIRplus project, and we will continue to work on going forward. So yeah, that I hope I answered Anne's question and yours, Victoria, at the same time.
Yeah, I can add to that a little bit. So I mean, I think one of the things to really capture here is the fact that often people, when they're generating their data, don't realize the impact that it can potentially have on communities that they're not used to working with. So people generate data for their specific scientific research question, but don't realize that in aggregate with lots of other data sets, that can actually answer a fundamentally different biology question. And this is really where what we put a lot of effort into about standardizing and richly describing your data set so that others can come along many years down the line and pull your data together with other data sets to answer a different question. And a good example of this is some of the recent farm GTEx projects, where they're sort of bringing together lots of different animal agricultural data sets from around the world to sort of generate a tissue by expression atlas. So getting tissues from lots of different projects from all around the world. And they're able to do this because these data sets have been openly provided to the community, and also because they have really rich and fair metadata descriptions. And what this means then is that they've then be able to generate this sort of larger resource that can then tackle questions that the original researchers themselves were probably not thinking of focusing on. And really in the biodiversity context as well, this is really important about sort of ensuring that in the countries that are sort of most advanced in the biodiversity projects, as the Darwin Tree of Life project, that we're then able to sort of share our expertise with other countries around the world, because we need to sort of build that capacity in countries. And this is what Thank God is really working very hard on, is to ensure that those own countries can then sequence their species, because if we're going to get a complete picture of the whole of the Earth's sort of biodiversity, we need to be able to sequence in all of the countries in the world, and not just in the ones that are particularly well funded or have particularly sort of well-established sort of scientific bodies. And that's really what we need to sort of try and continue to support, and to continue to support those countries to sequence their own data.
Yeah, so maybe I can just add more to that. So just as Peter mentioned, there's a wealth of knowledge to actually that we can actually benefit from if we make this data open and then pull resources and ensure that the data is open. But as I mentioned during my introduction, is that one of the things basically is that sometimes the data is open, and which is something that happens quite often. And one of the things is that, but sometimes the capacity to actually utilize the data is not there. So one of the things we tend to see is that communities tend to kind of withdraw or try not to make it open, in the sense, because the question they ask is, okay, I make it open, what do I benefit from this? If there's an IP that comes or a patent that comes from this in 10 years and 5 years, how do I benefit from some of this? And I think those are some of the questions that keep coming up, basically, even while trying to engage the public on the African Biogenome Project. So, and it is one thing that keeps coming up. And one of the things we're trying to do, and I say it again as a long-term thing, both in the short term and long term, is the capacity, trying to build the capacity. And it is the fundamental aim of the Open Institute, to be able to build that confidence and say, we're going to do this on the continent. This project is going to build capacity. And of course, because most of the custodians of these samples are indigenous peoples and local communities, and we try not to go to people's, for instance, backyard or the farmland and get biodiversity and sequence. It's something that we don't do. So that is some of the things that we tend to see around here, the project basically.
And I guess one other kind of angle that I will bring to that is, if you're thinking about kind of how industry would maybe work with data, and there's a lot of proprietary data that's about kind of making your product, but there's usually this kind of pre-competitive stage where everybody kind of can work on the same thing without kind of harming anybody else's kind of, plans for their production. And so pooling resources and coming together and doing that to kind of generate those shared baseline resources that then everyone else can work from and save everybody having to do it independently. I mean, that's really a lot of what this is about, kind of data reuse as well, right? Just to bring costs down and make sure we can all kind of move faster and get to our goals kind of in a more efficient way.
Great, thank you all so much for sharing your perspective. So now we would like to shift the focus to audience questions. So first I want to ask, so there's a question for Thank God, which is, what has the reaction been so far from the scientific community in Africa and outside when you've talked about the African Biogenome Project? And what are the things that people are getting excited and worried about? And I guess also I'd like to extend this to other panelists too. What are some aspects of the excitement, but also the challenges that you have to face? So perhaps first, Thank God.
Yeah, so I think the excitement about the project, which is it kind of, when we did announce the project a few months ago, there was huge interest from the continent and from the early career scientists and from other scientific agencies, and they see this as an opportunity for the continent to build its own capacity. One, they see this as an opportunity for the continent to better understand its biodiversity. Two, they see this as an opportunity for the continent to have data-driven scientific evidence to be able to engage conversations on the post-2020 biodiversity framework and other, the Convention on Biodiversity and, just about all the biodiversity activities. So those are some of the excitement that comes with it. But again, within that excitement is, okay, the continent does not have as much capacity as most continents. For instance, the continent does not have a central database to store its own data or process its own data, and the continent does not have central locations to sequence its own genomes. And just about 1 or 2 countries, for instance, South Africa and Nigeria, and maybe Egypt. Then how is that going to be done? Because some of this will require interacting with communities. And one important thing is that in assets and benefit sharing negotiations, because the assets and benefit sharing basically is more of a protocol that was agreed in Nagoya to kind of try to look at the benefits that arise from biological resources, and of course biodiversity is one of them. And usually you have to have a negotiations in that process to be able to use community samples. So and in those negotiations, capacity building is one key criteria. So for instance, if someone in Gabon, for instance, a scientist who maybe have a sample and someone in Switzerland wants to collaborate, and then there could be a negotiation that says, okay, I could be trained in genomic sequencing or genome sequencing or biodiversity conversation. Of course, that could then be a form of negotiation and being able to share the benefits that arise. So those are some of the things that began to come up, and they again form the basis on how some things have been evolving within Africa BP on how can we address some of those things while at the same time promoting open science and ensuring that some of this data is available on the databases as well.
Yeah, extending this question to other panelists, do you also encounter this dilemma when it comes to being open as much as possible, but also making sure that there are certain their benefits are being shared by the producers and the users of this data?
So I think this is a hugely interesting area at the moment that's really getting discussed a lot around sort of the access and benefit sharing in particular countries and Nagoya protocols, and really about how we can ensure that the countries themselves are able to then benefit from any discoveries that are made from particular data sets. And that's it's a really key part of sort of balancing against fully open data, because it's open data with the context that we need to ensure that the communities that are providing these resources are able to benefit from them, so that the scientific community can still move forward, but then that if there is sort of a derived financial or benefit from that is shared with the country, that it isn't sort of seen as just being taken away from them. And I think that's a really area that's being a lot of thought being put into at the moment in the context of global biodiversity projects. And there's really a lot of discussions going on globally about how to best manage this. And I think really that is an area that continues to need really careful thought and consideration and partnership between different projects and communities, which is why the European Bioinformatics Institute has sort of signed a memorandum of understanding with the African Biogenomes Project to really try and support them in building their own capacity and providing our training and expertise whenever we can to help them be able to build that capacity within the continents to then sequence their own genomes, to then distribute them openly wherever possible and support them in their scientific endeavors that they want to do.
I think I would add similarly, when we're talking about biodiversity, we're maybe thinking about the breadth of species here. But when you're thinking more kind of within a crop, you might be talking about the different varieties or traditional varieties that have been grown, the land races, on their wild relatives, and then how they can be kind of bred in. And again, this raises the whole issue of benefit sharing, and how to do that in an appropriate way that's kind of gives back. And so there's a lot of an awful lot of discussion around this, the generation of digital sequence information and how that can then go on and be used, and how that kind of can feed back in a fair way those benefits. So it's definitely is a big area, lots of people thinking about it, very important and important to get it right.
Yeah, so a related question that Anne asked, in your opinion, does industry share as openly as academia? Is open sharing across communities balanced? So maybe Tony can take this one also, because you work with some industry partners as well.
I can start, and I think Sarah will have opinions as well here. So I think it's a really good question. I think I pointed a little bit at this about FAIR and is not necessarily open. I think there's plenty of occasions that we see in industry where industry are more interested in sharing certain proprietary data assets, and Sarah mentioned the pre-competitive component that exists to start with. I think in general industry is moving in a very good direction in sharing pre-competitive data, but most of the industry partners we work with are very sensitive about sharing openly sharing any data that they may conceive as conveying some sort of competitive advantage. So I think the answer is mostly no, but with justification. And I actually think industry is doing a really good job of moving in the direction of ensuring that their data might be fair, but not necessarily open. So data sets that maybe have been mined for what they think is their competitive advantage might be released in an interoperable way in future. So I think there's this component of sort of, obviously industry want to protect their investment in certain sort of anything that will give them a competitive advantage, and that makes sense. So fair is good, maybe not so open. The component about open sharing across communities being balanced, I think that's definitely not the case. Some communities are much further along in their best practice in terms of openly sharing data, and it's not, I think most of that is not related to sort of a desire to hold on to data or protect it, but just that there are certain communities that we work with that maybe aren't as far along the process of open data sharing as others. I think that here at EBI, we have this kind of quite privileged view of most of the people that we work with in biosciences are sort of fairly far along that journey of open sharing, but there's definitely communities where the practices of open data sharing is very new to them, and there's huge opportunities for knowledge transfer from the work that we do into other communities in those cases, I think. So yeah.
I think I would have to agree with that. It's if we think about kind of how industry sharing data, that's also quite different across different areas of industry, and as we sort of mentioned, this pre-competitive space, and there's some very nice examples where the Open Targets project is a big one, which I'm not directly involved in, but is worth mention here, where industry is coming together to work to kind of bring together human data for clinical purposes, but again, in that pre-competitive space. So they're developing a platform together, that they're all working together to kind of do that, so that work doesn't need to be done multiple times, which is really fantastic, and hopefully will kind of be a model for other communities moving forward, which may or may not look exactly like that, but the idea of kind of working together to produce something that will benefit everybody, and kind of, yeah, help them along in the process.
Yeah, so just to also add to that is that Anne's question about, you know, the industry not being open. I think it's not just the industry as well. We see that in other communities that do have private servers, for instance, and you know, they're out there in terms of being, you know, having it in private, and most of them are not, again, people in the industry. So it's all about being able to see how do we integrate all of these communities and also sequences, basically.
Thank you. So there's one more question regarding if there's any grant or funding challenges. Also, would just like to tie this into maybe a larger question, which is what are the major challenges? Grant funding, were other ones in your project, and how can we overcome them?
I mean, I think funding is always a huge challenge with these endeavors, particularly when you're sort of going for these sort of very large, long time scale projects where you're trying to, for example, the Earth Biogenome project, where you're trying to sequence the whole Earth's biodiversity. These are hundreds of millions of pounds worth of projects. And the way that this particular approach is being done, and rather than have a single entity to get that much funding into a single project would be incredibly difficult. But the approach that's being taken is actually that there is, rather than a single entity, it's actually a collection of, so far, around 60 smaller biodiversity projects that are either at a country or a continent or focusing on a particular scientific clade. And really that way, by them coordinating across those, you ensure that they are still comparable, that the data set produced in the US is as comparable in terms of quality and metadata descriptions and richness as the data set produced in Europe. And I think that's the sort of, for some of these really large scale projects, it's sort of the only way we can really tackle the sort of the financial scale that's needed to sort of do this. I mean, unless there would be a sort of a coordination of funding across sort of all of the other countries, or particularly sort of wealthy individuals or industries, then I think sort of this coordinating across multiple projects approach really can help a lot in these situations.
Yeah, just to add to that as well, and the funding is always an issue, as Peter says, and actually the challenge, I think, is that a lot of what we talk about in this space, this kind of data management, open data sharing, data coordination, really requires a lot of investment in good data management, and that stuff is kind of more difficult to fund, because I mean, all funders want to fund the science, right? They want to fund the scientific impact, and if you you know, Peter said it very nicely earlier, that actually a big slice of this is actually making sure that your data is reusable for potentially other scenarios that you didn't imagine when you generated the data in the first place. And that comes with a little bit of cost, right? That nobody particularly wants to fund, because it isn't directly related to sort of an initial scientific endeavor. So these kind of like just good practice things generally tend to be a little bit hard to sort of explicitly fund. But I also think, you know, there's a movement towards a recognition that actually good data management, open data, open science is really, really important. And you know, I think we're gradually realizing that actually we should be investing in managing the data assets that we collect and that we generate. So it's not a solved problem by any means, but I think we're moving in the right direction, where funders are gradually recognizing the importance of good data management. Most grants that we write now require sort of a good data management plan, for example, and these things are sort of growing and gaining traction. So I think we're moving in the right direction, even if we're not there yet.
Yeah, just to add to what Tony and Peter said, I think another aspect that is challenging is the sample collection. There are more samples in the wild than there are in museums and zoos and other places. And if we want to sequence all of those, then there has to be some collection mechanism and metadata that accompany those data, because that is the only way whereby we know that an accession in ENA goes back all the way to a particular sample collected somewhere. And I think that is one aspect that I think is a bit challenging. And of course, along the value chain of the sample collection, then there is also other elements in terms of species crossing across multiple countries, and each of those countries having different legal framework on how you deal with species and how you deal with, you know, the sequences and just about everything. I think for us, I say the sample collection being one major challenge, and because there are more species out there in the wild than there are in museums, because with the museum sample it's easy to trace, but with those in the wild, if you need to actually go collect them, put them somewhere, or then it becomes again a big challenge, basically.
Okay, great. Thank you so much for that. So I see that we have a few minutes left. Sarah, would you like to also comment on the challenges in your work with Ensemble, or?
Yeah, I suppose as has been alluded to, like which links back to the funding and many other things. The scale of what we want to do is, you know, we're very ambitious, and we want to bring in as many data sets that can be useful to people as possible, or support people to generate their own data and analyze their own data and do that kind of capacity building. But I think globally we're very ambitious. Everybody that wants to kind of, you know, as the cost of sequencing has come down, but our ambition has grown. So here we are looking at kind of wanting to do everything. So scale is something that we have to consider always, and how we prioritize that, which is often we have competing interest areas of priority. So, you know, we have to balance that, and I think that's true for everybody kind of generating these kind of resources or hosting resources. So yeah, that's definitely, I would say scale is up there as one of the big ones.
Thank you so much for that. So I'd like to just close off by thanking all of our speakers for sharing your work. It has been very insightful to hear how you support the adoption of open access to data and enabling research from very different angles. So we look forward to seeing how your projects grow and develop. And we hope that you keep the conversation going at EMBL and also in all of our project communities. So thank you very much for that, and hope you have a great rest of the day and Open Access Week. Goodbye.